Hello, thanks for the organizers to invite me, and thanks for attending this presentation. This presentation will contain forward-looking statements. So what do we do at Atamyo? We develop a new generation of gene therapies that harbor breakthrough technologies that improve dramatically the efficacy and safety of such gene therapy. And more specifically, we do gene replacement through AAV, and we have an initial focus on two families of disease: limb-girdle muscular dystrophies, LGMDs, and cardiomyopathies. And what differentiates us from many other companies is that this new generation gene therapy is being tested in clinical trials today and generating exciting clinical data. So we are a spin-off from an organization called Geneton, and thanks to this collaboration with Geneton, we have a hybrid business model. The first part is that we work constantly on generating new capsids, new promoters, and other breakthrough organ sparing technologies. And all this innovation fuels our clinical stage pipeline. So in green, you can see we have three LGMD programs, one generating exciting clinical data, an LGMD-R9, a second for which we already filed the clinical trial application, and then a third program that should enter clinic in 12 months' time. And we have now started a very exciting breakthrough programs targeting dilated cardiomyopathies. You can see on the right that each of these programs has an interesting commercial potential for LGMD. And for dilated cardiomyopathy, it's a completely different paradigm because we have identified ways to target with one single administration, one vector, several dilated cardiomyopathy, whatever the gene mutation is, and that is causing this dilated myopathy. So I have one slide to just illustrate some cool stuff being developed at Atamyo in collaboration with Geneton, the new capsid generation. So first to mention that with Geneton, we have set up a very efficient way to screen all these capsids, starting with a single cell and progressively testing these new capsids in human cell lines, and last step on proprietary in vivo models. And this allows us to screen very efficiently capsids that are being generated with machine learning. And there are three important originalities here that I disclose on the right. Is that first, we can do that. We have a very large library generated by Geneton over 20 more years. So we have real data with capsids in the magnitude of tens of thousands of capsids. And the second originality is that the algorithm predicts very well the manufacturability of these capsids. And the third innovative point of this algorithm is that the algorithm is set to predict how these new capsids can escape from pre-existing neutralizing antibodies. On the bottom right, you can see illustrations of how this algorithm is predictive. One point is a real data. So on the x-axis, you can see what is the data that is predicted by the model, and on the y, what is the real data. And you can see that, so to illustration, the productivity of this AAV generated, so this machine learning generated AAV, sorry, and on the right, expression of this induced by these capsids in Duchenne myotube. So this. Now let's focus on our pipeline. I will start with ATA100, which is our one-time gene replacement therapy for LGMD-R9. So LGMD-R9 is caused by a mutation of FKRP. The gene of FKRP we code for an enzyme. FKRP is an enzyme that is responsible for the correct binding of the muscle fibers to their environment. So mutation of FKRP leads to progressive muscle contraction-induced damages and muscular dystrophy. So in terms of clinical manifestations, almost all patients will lose at one moment the ability to walk, but it's very also important to mention that this FKRP mutation affects also the diaphragm, so leading to a fibrosis of the diaphragm. So these patients, the first symptoms will be the difficulty to breathe. There is no treatment approved, so high unmet needs. So in terms of vector, we built a best-in-class vector which harbors several breakthrough innovations. First, we have introduced a codon-optimized sequence that increased the efficacy by 5-fold. For the first time also, we introduced a sequence that targets very efficiently the heart. So we have a decrease of the expression of FKRP in the heart, so preventing cardiac toxicity. FKRP, the accumulation of FKRP in the heart, tends to induce very early on cardiac toxicity. So all this combined leads to have a vector that not only has outstanding efficacy, we have full restoration of histology and force at unprecedented low dose for a single administration, 9^12, but on top of that, we have an outstanding safety profile. I will come later to, just after that, to the clinical trial design, and it's important to mention that we have an outstanding manufacturing process thanks to our partner SK, and we already have a very cost-effective way to manufacture this gene therapy. So in terms of clinical design, we have filed, and we were clear both in Europe and in the US for all-in-one clinical trial. So it's one single clinical trial that encompasses both a dose escalation and a phase 2B, placebo-controlled, and it's designed to file for a clinical trial application after 1 year follow-up of patients in the phase 2B. We have decided to conduct this trial in a very homogeneous population, defined as patients that have already a respiratory impairment. We want patients that still are able to walk because we are documenting how our treatment improves the walking functions. And for the phase 2B, we will add another criteria, is we want to have patients that have already a significant functional impairment measured by NSAD, so an NSAD inferior to 40 over a maximum score of 54 for healthy subjects. In Europe, the primary endpoint was accepted as being a respiratory endpoint, so FVC, the difference at 1 year FVC between treated patients and placebo group. And in the US, after very constructive discussions with FDA, we were clear to have a combined endpoint that is a combination of the respiratory endpoint, FVC, and improvement in NSAD. So we have those first 3 patients, and I'm a bit frustrated. I cannot present you the results because I am embargoed. We are presenting the results in 2 weeks' time at the DHECT, but for the first 3 patients, I can tell you that we are very happy because these first results are absolutely in line with what we have seen in preclinical models. So I invite you to attend this presentation at DHECT, and we'll do a press release in 2 weeks' time. So that's for the lead program. The second program is very interesting. So we are proud we announced a few months ago that we have filed a CTA for this program. We are the only company to develop a treatment for LGMD2I. It's called by a mutation in gamma-sarcoglycan, who is also responsible for the correct binding of muscle fibers to the environment. In that case, the clinical manifestations are much more aggressive. The patients start lose the ability to walk from 5 years old. All patients will lose the ability to walk at 15, and half of these patients are severely cardiac impaired. And what's I want to give this example. What's very unique is how we chose the promoter. So building next generation gene therapy, it's not only about capsids, it's also about promoters. On the left, you can see the efficacy. On the top left, normal muscle, muscle of a normal mice. On the top right, the same muscle of the mice that carries the same mutation than patients. And you can see that after 1 single administration, 2 months later, we get back to normal histology. So that's for efficacy. Safety. For this disease, and that's the thesis that we implement in all our programs, we want to have the same biodistribution and in healthy subjects. Gamma-sarcoglycan, you have the same level of gamma-sarcoglycan in heart that in skeletal muscle, TA, tibialis anterior. First-generation gene therapy tended to have an overexpression in heart, such as driven by promoters such as desmin and CK8. You have 40 times, 30 times more your protein in the heart than in the skeletal muscle. This leads to cardiac toxicity. So we chose this promoter to have exactly the good balance. And the good surprise is that in top of that, this promoter targets very efficiently the liver. So for this program, first time in my 20 years career of biotech, we have never been able to find a dose leading to toxicity. We went up to 50 times the dose we want to inject in patients, never have any adverse event in preclinical models. So now we let's just one slide to describe a very original program in cardiomyopathy. So what we this is the Isabelle Richard, our chief scientific officer. She is the one who sequenced a TTIN gene many years ago, and her objective was to find a way to address dilated cardiomyopathy, whatever the gene mutation is, because TTIN, for example, cannot be encapsulated in a AAV vector. So she did an analysis for several different dilated cardiomyopathy, and she found pathways that are common to these different dilated cardiomyopathies. And here you can see an illustration of proof of concept. Is the same vector that is able to prevent the development of cardiac fibrosis on the left in a dilated cardiomyopathy caused in a Duchenne model, and on bottom right in a dilated cardiomyopathy caused by in a TTIN mutated model. So that's very original, and in fact what we are tackling is the formation, development of cardiac fibrosis. So that's what we are doing, and we hope to have vectors that could treat any dilated cardiomyopathy very efficiently. So what is next for Atamyo? Thanks to our pipeline, we have a very rich milestones that are being generated. For the lead program, we will continue to generate, it's an open study, the phase 1b, so we continue to generate efficacy and safety. We could start the pivotal phase second half 24. For the second program, we will dose the first patient early 24, and we'll generate in 24 safety and efficacy result. Third program that I did not present here, but it's the most prevalent LGMD caused by mutation in calpain. And we have a best-in-class vector, and we have started the IND-enabling tox study. We should be able to file the IND in 1 year time. And of course, we will focus the next 18 months in developing, in consolidating this unique position for dilated cardiomyopathy, not only in vectors, but not only regarding the targets, but building this new, generating these new capsids thanks to the machine learning engine that I showed you at the beginning. All this will be made possible with fundraising. So we have a original equity story. We have been funded so far only by our parent organization, Genethon, and its associated organizations called IFM, it's two patients associations. And we are starting, so the first fundraise, Series A, with clinical data in hand. And it's a 40 million Series A that we are raising. And we should be in 1 year time with a unique profile with 2 programs generating exciting clinical data, of which one with a clearer roadmap to being approved, to the approval, and a very unique position in dilated cardiomyopathy. That's it for today. Thanks a lot for attending my presentation, and I will be outside if you have any questions.